Right, today we are going to talk about gametogenesis. Gametogenesis is the process by which the early germ cells undergo process of meiosis and cytodifferentiation and eventually convert into mature gamete. So what is gametogenesis? Gametogenesis is a specialized process which occur in the germ cells, primordial germ cells and germ cells, that these germ cells, right, they undergo process of meiosis, right, and meiosis is a special type of cell division which occurs in two stages, meiosis I and meiosis II. And during meiosis, chromosome number is reduced to half in the final daughter cells, right? So meiosis is also called reduction division. Why we call meiosis reduction division? Because meiosis is a very specialized type of cell division in which after two stages of meiotic divisions, the final cells have their chromosomes number halved. Is that right? Or we call it reduction division. So what is really gametogenesis? Gametogenesis is a process in which the germ cells convert into mature gametes, right? Now, in females, gametogenesis is called the process of oogenesis, and in males, the gametogenesis process is called spermatogenesis. In females, during gametogenesis or oogenesis, what really happens that female germ cells pass through process of meiosis and then they undergo process of cytodifferentiation and maturation and eventually produce mature gamete, which is called mature ovum. Is that right? And in males, process of spermatogenesis, process of gametogenesis in males is called spermatogenesis. And during spermatogenesis, again, there is process of meiosis, which reduces the chromosome number half in the final daughter cells, and process of cytodifferentiation, which make the maturing germ cells in a final development of mature sperm. Is that right? Now, first I will go into general concept of meiosis. Meiosis is occurring in females during oogenesis. It is occurring in males during spermatogenesis. So I will discuss first of all meiosis in some basic detail, and after that we will discuss into detail what is the process of oogenesis in female, and after that we will discuss that how the spermatogenesis occurs in male. So let's first of all discuss about the primary basic concepts of meiosis. Now, during the process of meiosis, let's suppose these are early germ cells. For example, this is a germ cell. If it is in female, this must be oogonia, or if it is in male, then it may be spermatogonia, type B. Now, these are early germ cells, and in early germ cell, what really happens that, let's suppose if I'm going to make sperm, right, then what will happen in my early germ cell, which are spermatogonia, I have how many chromosomes? 46 chromosomes in the early germ cells, right? And these 46 chromosomes, 23 I have from my mother, right? And 23 chromosomes I have from my father. So basically, how many chromosomes I have? 46 chromosomes, right? So 46 chromosomes are there. 46 chromosomes are there. And chromosome, when they are 46, we call them their diploid number of chromosomes. What is this? Diploid. Diploid number of chromosomes. So what really happens that in early spermatogonia type B, we have how many, a male have how many chromosomes? 46 chromosomes, 23 coming, in my case 23 chromosomes must be coming from my mother and 23 chromosomes must be coming from my father. So for example, it is chromosome number 1. So this is my chromosome number 1 derived from my mother, and this is my chromosome number 1 derived from my father. So and so forth. I will have chromosome number 2 from mother as well as father, chromosome number 3 from mother and also one copy from father. So and so forth. So 22 chromosomes will be homologous to each other, because I have 23 chromosomes from mother and I have 23 chromosomes from father. These all 23 three chromosomes are homologous to each other. But you have to remember, X chromosome in my case must be coming from my mother and Y chromosome must be coming from my father. Is that right? Now what really happens? If you look at the genetic material, amount of DNA, remember when we are talking about cell division, we have to talk about what is the number of chromosomes and we have to talk about what is the amount of DNA in a given cell. Right? Now here the number of chromosomes should be measured by the number of centromeres. Right? Now in this early cell, before it enter into meiosis I, right, this early cell, Spermatogonia, the number of chromosomes is 46 and each chromosome has single structured. Let me explain this. Let's suppose this is chromosome from my father and here is chromosome from my mother. Let's suppose these chromosomes are homologous to each other. When I say these chromosomes are homologous to each other, it means that both of them are having alleles of the same genetic material. For example, if it is chromosome number 3 from father, paternal, then it must be chromosome number 3 from maternal side. And if there here is a gene in the DNA, of course, what is there? There must be double-stranded molecule of DNA. Every chromosome, look, this is double-stranded molecule of DNA, and if these chromosomes are homologous to each other, right? Homologous to each other mean chromosome 1 of mother is homologous to the chromosome 1 of the father. Chromosome number 10 coming from mother should be homologous to the chromosome number 10 of the father. Is that right? So corresponding chromosomes from mother and father are homologous to each other. X and Y are not homologous because they are not having the same genetic material. Right? Now listen. Now these are one pair of homologous chromosomes. Is that right? Now in this case, both chromosomes are having the similar DNA, and in the DNA there are genes, right? What are genes? Genes are sequence of nucleotides which make a specific functional RNA. Now let's suppose if there is a gene here, right, and this gene is about the color of the eye. Now this gene which is present over here, it determines the color of the eye, and this gene is coming from the mother. Then on the same homologous chromosome which is coming from the father, at the same locus, at the same point, there should be gene which also determines the color of the eye. And here is suppose another gene which determines the size of the nose from maternal side. Then on the homologous chromosome coming from paternal side, on the same locus, right, from paternal side, there should be gene, and this gene should be also determining size of the nose. For example, here there is a gene which determines structure and function of a specific enzyme, right? Then on the corresponding locus on the other homologous chromosome, there should be a gene which should be responsible for the structure and function of the same enzyme, right? Now these genes, look, these genes which are going to do similar function, and they are present on a homologous chromosome at the same loci, right? These genes are in specific relationship to each other. As I told you, this gene is making color of the eye, maternal side gene, maternal gene for color of eye. This is paternal gene for color of eye. Both of these genes are alleles of each other. What are they? They are alleles of each other. And what is this? Gene for the suppose size of the nose or shape of the nose from father side. This is the gene for the shape and size of nose from mother side. So these two genes are also alleles of each other. Then if this was a gene from paternal side for a specific enzyme, and here on the same locus on the homologous chromosome coming from the mother side also has the same similar gene for the same enzyme, then these genes are also alleles of each other. So what we can say, when two chromosomes are coming from mother and father, is that right? And they are having similar genetic material, such chromosomes are homologous of each other. And on the homologous chromosome, the genes which are having the same loci in their position and doing the similar function, right, or dictating the similar genetic information, these genes are what they called alleles. Is that right? Any question up to this? No problem. Now, you must remember also one more thing. These are the chromosome, let us suppose chromosome number 3 paternal. This is called single structured chromosome, right? This is also maternal chromosome, but this is also single structured. When chromosomes replicate their DNA, chromosomes become duplicated. Let's suppose this chromosome, right, it replicates its genetic material. When the DNA replication will occur, what will happen to this chromosome? After DNA replication, this chromosome will become duplicated. Is that right? In the same way, if DNA has been replicated in this maternal chromosome, this maternal chromosome will also become, yes, duplicated. Now, here it is very important, in duplicated chromosomes, right, of course, DNA material is double. Is that right? Because DNA has been, yes, replicated. Replication of DNA lead to duplication of chromosomal structure. Or we can use another term. Now listen. This chromosome, which does not have replication of DNA, this is called, it is one chromosome, one copy of chromosome number 3, and it is single structured. It is also one chromosome but double structured. Why? Because chromosome number is basically counted on the base of centromere. Is that right? Centromere is the point where the chromosome is constructed. Is that right? Now, so this is single structured chromosome, this is double structured chromosome. Single structured chromosome is unreplicated DNA, double structured chromosome has replicated DNA. Is that right? Single structured chromosome is also called chromosome with one chromatid, and this is also called one chromosome with two chromatid, right? Or we call this unduplicated chromosome, and this is called duplicated chromosome. Let me recap. When one chromosome has one centromere and one short arm and one long arm, what we call that this is unduplicated chromosome, or it is chromosome without replication, or it is single structured chromosome, or we can call it chromosome with one chromatid. Is that right? But if this situation is there, the DNA of the chromosome has been replicated, it is still one chromosome. So what we'll call this? One chromosome with replicated DNA, or we can call it one chromosome with duplicated structure, or we call it one chromosome with double structure, or we call it one chromosome with two chromatids. So I hope you will remember these terms. What terms I have made clear in your mind? What are the homologous chromosomes and what are alleles on each other? Is that right? Homologous chromosomes have homologous alleles, and then I talked about what are single structured and double structured chromosome. In my lecture, I will continue such chromosomal structure as single structured chromosome, and such chromosomal structure I will talk about double structured chromosome. Any question here? No. Now let's move forward. Let us suppose that in my body spermatogenesis is going on, and if spermatogenesis is going on, it means my germ cell must be undergoing the process of meiotic division. Which division? Meiotic division, which is a specialized type of division which occurs especially in formation of germ cells and gametogenesis. Is that right? Now, let's suppose this is in my testis. This is the primary cell. Of course, my testis will not have oogonia, right? Hopefully. Now we come to spermatogonia B. Now, spermatogonia B in my body, it has 23. Let me write here. It has 23 maternal chromosomes, 23 maternal chromosomes, plus it has how many chromosomes from paternal side? Also, yes, 23 paternal chromosome. Now, when there is a full set of 23 chromosome, we call it 1n. One small n. So we will use a term here that in this cell, so spermatogonia, there is number of chromosomes, one small n from which side? Paternal side, one small n from maternal side, right? One small n is equal to 23 number of chromosomes, one set of 23 chromosomes. Now, if you put them together, right, how many n's are there? It is 2 small n. And when there are 2 small n, it means it is double of 23. So we can also say that this 2 small n is 46 chromosomes. Or when number of chromosomes is 2 small n or 46, we say it is diploid number of chromosomes. What we call it? Yes, diploid number of chromosomes. I'm talking about this cell which is about to enter into meiosis I. Now, another thing, this was about the number of chromosomes. Let's talk about the genetic amount of DNA. Is that right? Now, amount of DNA, this is single structured or double structured? Single structured chromosome. And what is this? This is also single structured. Now come back here. In the single structured chromosomes, if there are 23 chromosomes which are single structured, 23 chromosomes which are single structured, we call it the amount of DNA is 1 capital N. Capital N is notation used for the amount of DNA in a given cell. Small n is the notation which is used to the number of chromosomes. Now, if we talk about there are 23 paternal chromosomes, single structured, what we will say? That there are 1n paternal chromosome and 1n mean 23 chromosomes. And I say 1 capital N also, right? This is not only 1 small n, it is 1 small n and it is 1 capital N. Now, when I say it is 1 small n situation, it means there are 23 paternal chromosomes. And if I say 1 capital N, it means it is single, all 23 chromosomes are single structured. What is this? Single chromatid or single structured or single chromatid chromosomes. Is that right? Now, in the same way, these 2 chromosomes I have drawn here, right? Again, it's maternal side. How many chromosomes from maternal side? There are again 1n from maternal side, so 23 chromosomes set from maternal side. And each chromosome is double structured, single structured here. Single structured, so it will be 1n, single structured. Is that right? So in this case, what we will say? That 1 small n plus 1 small n. So what is the number of chromosomes? 2 small n. Or we can say, what is the number of chromosomes total? 46. So this cell has 46 total number of chromosomes and diploid cell. But look at the DNA of it. It has 23 single structured from paternal side. It has 23 single structured chromosome from maternal side. So 1n from maternal side, this is amount of DNA. So amount of DNA from mother is 1n. Amount of DNA from father is 1n. So how many capital N amount of DNA? 2n. Is that right? Any question up to this? No. Now we come to this situation. Actually what happened, before this cell is entering into meiosis I, before this cell enter into meiosis I, it is going to replicate its DNA. And all chromosome, paternal chromosome as well as all maternal chromosomes, they will replicate DNA. When all the DNA is replicated, all the chromosomes will become double structured chromosome. What they will become? Double structured chromosomes. It means when these single chromosomes will become double structured chromosome here, so these structure will convert into this type. So it means every chromosome is a double structured. Now this cell with double structured chromosomes, right? This is called GS. What is this called? It is called primary gamete. What is it? What it is called? Primary gamete. Of course, this primary gamete in female it will be primary oocyte, and of course I'm not female, so it will be primary spermatocyte. Is that right? Now, in the primary spermatocyte, which is derived from Spermatogonia B, or in the female, primary oocyte, which are derived from Oogonia, right? They are having double structured chromosome because they have already replicated their DNA. And if they have already replicated their DNA, now very carefully we'll talk about the structure, right? How many paternal chromosomes are there? Now we have 23 paternal chromosomes but double structured. Here were 23 paternal chromosome is single structured. So what we'll say that paternal chromosomes are 23. How many paternal chromosomes? Paternal chromosomes are 23, and how many n? 1 n, one small n. Is that right? And plus on paternal side, maternal side, yes, maternal are how many? Look, at this cell there were 23 single structured, and here there are 23 double structured. So we'll again write 23 but double structured maternal chromosomes. Is that right? And how many small n it is there? It is still one small n, because even though they are double structured, but actually the number of chromosomes depends on number of centromeres. Is that right? So how much it will make? Both of them together. Total chromosome will become 2n small. Or we can say total number of chromosome will be, yes? No, it will be still diploid. This cell in the beginning will be still diploid. But there's a very important thing. Amount of DNA has been doubled here, right? Because every chromosome from here to here or from here to here has become double structured. Now, if we are talking about the amount of DNA, now here the amount of DNA was 2n. 1n was contributed by father and paternal chromosomes, and 1n was contributed by the maternal chromosome. Is that right? And in the same way here now 1n has become 2n. So 2n in paternal set of 23 chromosomes. So we can say that paternal this is actually 2 capital N amount of DNA. Here it is how much? Again 2n, because it is also double structured. So total n will be how much? 4n. So we can say that primary gametes mean in males primary spermatocytes and in females primary oocyte, right? All the chromosomes from maternal and paternal side are double structured chromosome, but number of chromosomes still remain diploid. Is that right? But amount of DNA has become double. Is that right? Any question here? Right. I'm just going to take your test because this bothers a lot to the students. Right? This concept, and then we'll move forward. Right? Yes. This is the early germ cell, right? How many? It is how many number of chromosomes? 2n. Right? 1n from father, 1n from mother. This is the number of chromosomes. So it is equal to 46. Right? It is equal to diploid situation. Is that right? But we talk about the, what is this? Genetic material. From mother, the amount of DNA is 1n, 1 capital N from maternal side, plus 1 capital N from paternal side. So amount of DNA is 2 capital N. Any question here? No problem. Now this cell convert into primary gamete, right? But before that, this cell has replicated its DNA, and every chromosome has become double structured, all maternal and paternal chromosomes. Now number of chromosomes. Now number of chromosomes remain the same because centromere is the same, but each chromosome has become double structured or duplicated or double chromated chromosome, right? So what we will write here? Number of chromosome 2 small n is equal to still 46 is still diploid. Is that right? But the real thing to understand here, amount of DNA has been doubled. Because amount of DNA has been doubled, so this was amount of DNA here was 1n and 1n. Now it has become 2n and 2n. So what is the amount of DNA? Is equal to 4n. Any question here? It's clear. After these some fundamental concepts, now we come to some special processes which occur in meiosis I. Right, there are some processes which occur in meiosis I, and these processes do not occur during mitosis. What is that? Listen. The homologous chromosomes in your cells, homologous chromosomes which are from mother and father, they are lying in the nucleus randomly. They are lying in the nucleus randomly, right? For example, chromosome coming from your mother, chromosome number 7, and chromosome number 7 coming from your father, they are lying in your nuclei randomly. Is that right? When mitosis occur, during mitosis now, all chromosomes replicate their DNA and become double structured. But double structured chromosome during mitosis, the homologous chromosome don't come near to each other and don't pair with each other. What does it mean? That let's suppose if in my body a skin cell is undergoing the process of mitosis, then chromosome number 7 from my mother and chromosome number 7 from my father will become double structured during mitosis, but they will not come close to each other. They will never pair with each other. They will never talk to each other. They will never, you can say, exchange material with each other. But during meiosis, something very special occurs. What is that? That is that homologous chromosomes from maternal side and paternal side, they come together and pair with each other. Right? The special thing is the pairing occur. Pairing means that, let's suppose this is maternal chromosome number double structured, chromosome number 1. Here is maternal chromosome number 2. Here is maternal chromosome number 3. You will be so surprised. You should be really surprised and appreciate what goes on at biological and molecular level, that maternal chromosome number 1 will look where is the paternal chromosome number 1 in the same nucleus. Homologous chromosomes from mother and father start coming together. Look where the other partner is. So what really happens that at this stage, at next stage, these double structured homologous chromosomes look for their homologous partner. So chromosome number 1 from mother will pair itself with chromosome number 1 from father, and chromosome number 2 from mother will pair itself with the chromosome number 2 from father, right? Paternal, and chromosome number 3, which is mother, it will pair itself. I don't know how they really look, but anyway they do it, right? So chromosome number 3 from paternal, and so forth. This pairing continues from homologous double structured chromosome to the homologous double structured chromosome from 1 to 22. Is that right? All the autosomes. And in the end, of course, if it is going on in female, it will be X and X chromosome come together. But if it is in male during spermatogenesis, then X from maternal side and Y which was coming from my father, they will also come together, and they have few homologous genes, right, on short arms, right? So what really happens that maternal homologous double structured homologous chromosomes during the meiosis 1 and paternal, now here meiosis 1 I'm going to explain up to this. Here I will come to meiosis 2. So first stage of meiosis, chromosomes maternal and paternal are already double structured. These homologous chromosomes look for their homologous partner, and every homologous chromosome pair itself, right, pair itself lengthwise point to point with the homologous chromosome. Why they are coming together? They have to do something. You know, they are going to talk and make special work for the next generation. Remember, in your somatic cells, maternal chromosomes and paternal chromosomes even don't talk to each other. They are randomly lying in the nucleus, but doing their function. Only during gametogenesis, it's a matter of next generation. So in my body, chromosome from my mother and father, chromosome 1 from my mother and father, they will come together. And in my wife's body, during oogenesis, chromosome number 1 from her mother and chromosome number 1 from her father will come together. Is that right? And what will happen then? Those chromosomes which come together and make pairs, right? This pairing, point to point, making the pairing, this process is called, what is this called? Synapses. So what these chromosomes are doing? They are making point to point, they are matching. So we call this is a process of synapses, or process of pairing of the homologous chromosome. After that, what they do? These chromosomes, once they have made pairs, you know what they do? They start exchanging the blocks of genes with each other. They start exchanging the blocks of genes with each other. Let me enlarge this for you. For example, this pair I enlarge. And what really happens there? This is maternal chromosome number 3, and now paternal chromosome number 6, they will become very intimate, you know. I don't know what they are doing at molecular level, but what biologists claim, now what they are going to do, they will exchange the blocks of genes. You know, one chromosome has thousands of the genes, right? Of course, this is having DNA molecule, it has also DNA molecule. These DNA molecules will come together, of course, as chromosomes come together, and they exchange the alleles with each other, right? And when they are exchanging the alleles and they are exchanging the blocks of genes, we say they are crossing over. So it's not only mother and father cross, even for the next generation in our body, when gametogenesis is going, homologous chromosomes also cross with each other and exchange their genetic material or shuffle their genetic material to add the diversity to the future generation. Now, so this process where they come together and eventually start crossing the material or exchanging the genetic material with each other, which I've shown here, this process is called crossing over. So crossover occur. Remember, this crossing over or exchange of genetic material or blocks of genes between the two homologous chromosomes especially occurs in meiosis. It does not occur in mitosis. Is that right? Now, how many such couples will be made there? 23. Is that right? How many exchanges will be going on and how many chromosomal pairs? 23 pairs. All pairs will be exchanging some material with each other. They say somewhere 30 to 40 crossover occur, right, in whole the maternal and paternal set of chromosomes. Do you understand it? So after the crossing over has been done, they will become separate, but they have exchanged some genetic material. And when they will become separate from each other, look, now they are going away from each other, but look here, what has happened? One piece of, yes, paternal, one piece of paternal genetic material has been exchanged with maternal chromosome, right? So now this maternal chromosome has some paternal genes and this paternal chromosome has some maternal gene. So this maternal chromosome is after the crossover, when they separate from each other, maternal chromosomes are not purely maternal and paternal chromosomes are not purely maternal, they do have some maternal material. And maternal chromosomes are not purely maternal, they do have some paternal material. Do you understand it? Now when these chromosomes will go away, they have made a unique recombination. Right? This was maternal genetic material, this was paternal. They come near, make synapses, and then they cross over and exchanges, and when they are separating from each other, maternal chromosomes have recombined their material with some of the paternal and vice versa. And when they are separating with each other, they make this from crossing over point, they are still attached, but rest of the chromosome couple is going away. Right? This situation is called formation of chiasma. What it is called? Formation of chiasma. So these are the very special and unique features which occur in meiosis, first meiotic division, which do not occur in mitosis. What are these special? Number one, process of synapses, where homologous chromosome pair with each other. Secondly, process of crossover, where homologous chromosome exchange the blocks of genes with each other, right, and to add the genetic diversity for the future generation. And then when they're separating under the microscope, they look like a very special arrangement, right, crossing each other, and when they're getting separate from each other, this is called chiasma. Is that right? These three features are not seen in, yes, mitosis. Any question here?
Even if the parents are divorced, these things happen?
Even if the parents are divorced, this thing will happen. That's the beauty. The molecular level, we are more committed than the social level. For example, my mother and father, if they are divorced, it doesn't matter. Even if they have died now. But in my testes, the chromosome which came from a mother and chromosome came from a father, they are still today, right, talking to each other before they make the sperms for the next generation. Is that right? Oh yeah, chromosomes don't fight. Yes, that's right. Chromosomes don't fight. Right? Now, listen. What we have done, we are talking about first meiotic division. We started with the primary gamete in males with primary spermatocytes and females with primary oocyte, right? DNA material is double structured. Chromosomes are 46 double structured chromosome with 4n material, 4n genetic DNA. Then what happened? Homologous chromosomes make pairs. Then they cross over, synapses and crossover, and eventually they separate, but now they have exchanged some material with each other. And what is this going on? Chiasma. And next step is alignment, that these chromosomes will align themselves on spindle, right? So next step is alignment. Alignment. Now, at the time of alignment, how many sets will align on the chromosome, on the spindle? 23 double structured which were originally maternal, and 23 double structured which were originally maternal, and 23 double structured which are originally paternal, right? Of course, they are now having some recombination. Then what will happen? These maternal and paternal 23 chromosome, right? They will be assorted randomly and going towards they will go away towards what is there, to daughter cell. Now here it is very important. Let me tell you. When these chromosomes are going away at this stage, for example, they are now going to assort suppose this is chromosome number 1, here is the set of chromosome number 2, here is the set of chromosome number 3. But of course you should not forget that there are some crossover already going on. Is that right? Now the maternal are not purely maternal and paternal are not purely paternal. Is that right? Now, for example, this is chromosome number 1 set, this is chromosome number 2, this is chromosome number 3. When they will move, as here they are moving, there are 23 sets paternal and 26 paternal. But look, for example, this is first daughter cell and here is the second daughter cell. First daughter cell may get chromosome number 1 from mother, chromosome number 2 from father, chromosome number 3 from mother. So they are randomly getting maternal or paternal double structured chromosome. And cell number 2, daughter cell number 2 will get chromosome number, double structured chromosome number 1 from father, 2 mainly from mother, and 3 mainly from father. So what really happens? That when after alignment, right, the chromosome will, double structured chromosome will disengage with each other. These homologous chromosome, what is this? Disconnect from each other, right? And then they, right? Now this disjunction, what is this? When they disjunction with each other, right? When they disjunction with each other, they are randomly assorted and allocated to the daughter cell. They are randomly allocated. It's quite possible this daughter cell number 1 here may have maternal chromosome number 10 with some paternal material and paternal chromosome number 11 with some maternal material. Is that right? Did you got it? So that will another way to add the genetic variability, right? Due to random assortment and allocation of double structured homologous chromosomes which have gone under the process of crossover. So up to now, by 2 mechanisms, genetic variability is occurring. Number 1, exchange of genetic material in between the homologous chromosome. Secondly, random assortment of homologous chromosomes to the daughter cells. And eventually, when daughter cell is produced here, at the end of the first meiotic division, these 2 daughter cells are called which daughter cell? These daughter cells are no more primary gamete, they are now called secondary gamete. What are these? Here we had, yes, primary gamete. Here we will have secondary gamete. Now, in secondary gamete, of course, either it is in males it will be secondary spermatocyte, and in females it must be secondary oocyte. But in case of female, this will become secondary oocyte and other daughter cell will get very little cytoplasm. So other daughter cell is called first polar body. In males, both of them will be called secondary spermatocytes. Secondary gametes. In the male, they are secondary spermatocyte. But in females, there are two cells which result as secondary gamete. One cell which get almost all the cytoplasm is secondary ovum. And other cell which gets very little cytoplasm, that is called first polar body. Is that right? But it's got a complete set of 23 chromosomes. Right? Now, let's see what is the situation there after the division. Now, this cell, again remember, when disjunction occur here, sorry, it was not non-disjunction, when they separate from each other, this was disjunction. When disjunction here, centromeres don't divide. These are the homologous pairs which go from each other, right, to the daughter cell. Homologous chromosomes pair which go to the daughter cell randomly. Now, in this cell, secondary suppose gamete, it is 1N. What is that? 1N. But it's single structured, double structured. It is double structured. 1N. For how many double structured chromosome? 23 chromosome, but double structured. Right? This should be called now, yes, this should be called 1N or 23 chromosome, but this is double structured, so we can also call it haploid now. Haploid number of chromosome. Is that right? So what we can say, in the same way, this cell, secondary gamete, it has how many chromosomes? 23, but double structured, right? So it is also how much now? 1 small n, 23 chromosomes, double structured, and haploid number of chromosomes. But amount of DNA is double. Is that right? Because every chromosome is double structured. Is that right? So what we'll call it? It is 2N DNA, right? 23 double structured. Here is also 2N. So what we'll say? This is 23 chromosome. Each chromosome is double structured. Due to that reason, it is one small n, right? Or it is called haploid. Is that right? Haploid. But when you talk about amount of DNA, it is 2N. Why it is 2N? Even though chromosomes are 23, you may think it may be 1N, but because every chromosome is double structured, so double amount of DNA, so it is 2N. Is that right? Now here I want you to be very clear. When the beginning of the meiosis, primary gamete are at the beginning of the meiosis, and one primary gamete gives secondary gamete at the end of first meiotic division. At the end of first meiotic division. In the beginning, how many chromosomes were there? 46 double structured. So they were diploid cells with 4N. Here are 23 but double structured. So haploid number of chromosome but 2N. Is that right? So this complete our first meiotic division. After the first meiotic division, these cells do second meiotic division, but when they directly enter into second meiotic division without replicating their DNA further. So these 23 chromosomes which are double structured, at next stage, just their centromeres disjunction. What breaks down? Splitting of the centromeres. And when their centromeres break down, each chromatid go to other side. This was double structured chromosome, and out of every double structured chromosome, one structure go to one side and other structure go to the other daughter cell. Right? So it means double structured chromosomes or chromosome with two chromatid split at the centromere point, and here centromere breakdown, right? Here only homologous chromosomes were going away during meiosis I. In meiosis II, centromeres will break down and double structured chromosome, one double structured chromosome will convert into two single structured chromosomes which go to the opposite pole and go to the daughter cell. Now, how many single structured double structured chromosomes were here? 23 double structured. They will eventually break down into 23, 46, what? Single structure. 23 will go on one side and 23 will go to other side. And same will occur with the other cell also. That this number, which is haploid cells, right? But double structured, they will again break down in the centromere, and single structured chromosome will go to the ends. Now, these are the final product of second meiotic division, right? At the end of the second meiotic division, right, we were having in the beginning of second meiotic division, we were having how many secondary gamete? 2 secondary gamete. And how much final cells are produced? 4 cells, right? Now, in this case, these 4 daughter cells which are produced at the end of the second meiotic division, every cell has how many chromosomes? 23 chromosomes. So every cell has 23 single structured, 23 chromosomes which are single structured, right? So it should be 1 small n. Or we can say this number is haploid. What is it? Haploid. But if you talk about amount of DNA here, how much is that? 1n. Is that right? So in all these 4 cells which are produced as a result of the second meiotic division, all these 4 cells will have haploid number of chromosome. Every chromosome is single structured, but every chromosome may be slightly different from other chromosomes due to crossover and random assortment, and amount of DNA is 1n. Is that right? Now, advantage of this is by the end of the meiotic divisions, first and second, every germ cell, sperm or ovum, is having haploid number of chromosomes. So my sperm will provide haploid number of chromosomes, and my wife ovum will also bring haploid number of chromosomes, and when they will fuse together in the zygote after fertilization, zygote formation will be re-establishment of diploid number of chromosome. Am I clear? Any question here?
Do you mind drawing that for a female ovum?
Yeah, what is that? At this stage, what happens to female oogenesis or spermatogenesis? Let me explain. In spermatogenesis, what happens? That one primary, in spermatogenesis, one primary gamete, which is primary spermatocyte, divides at the end of first meiotic division into 2 secondary spermatocyte, and then each secondary spermatocyte divide into 2 spermatid, which eventually mature into 2 sperms. So because 2 sperm will come from this secondary gamete spermatocyte and 2 will come from this, so in male, from one primary gamete, how many secondary gamete are made? 4. All of them become mature sperm. But in female, there is a little difference. The difference is that one primary gamete, during first meiotic division, when it makes 2 cells, one cell will get all the cytoplasm, and this cell is called secondary oocyte. Other cell will get very little cytoplasm, and that is called first polar body. Then in female, when secondary gamete is going to be mature ovum, then secondary oocyte divide into 2 cells. One cell takes all the cytoplasm. So this cell with 23 single structured chromosome and all the cytoplasm, this is called definitive ovum or mature ovum. And other is called second polar body. Is that right? Which is the second daughter cell, which is getting almost no cytoplasm. Meanwhile, this first polar body also divides into 2 cells, and they are also called third or fourth polar body. So we can say that actually what happens in female, out of the 4, what is this? 4 final daughter cells, 1 become definitive ovum and 3 are polar bodies, and the difference in polar body and definitive ovum is definitive ovum gets almost all the cytoplasm, polar body get very little cytoplasm and they degenerate. Is that right? They are not functionally useful, right? Now, if someone asks you, what is the purpose of meiosis? Meiosis is a very specialized type of cell division which occur in the germ cell to make the mature germinative cells. In males they make sperms, in the females they make ova. Is that right? During the meiosis has 2 stages, first stage and second stage of meiosis. Is that right? Now, during meiosis, during first step of meiosis, some special events occur. Number 1, homologous chromosomes pair with each other, synapses. Then homologous chromosomes cross over and exchange genetic material, and then they're separating from each other and making structure of chiasma. And eventually they align themselves and they separate from each other, but remember the centromeres don't break down. This is first meiotic division. In the beginning of the first meiotic division, we have which cells? Primary gametes. At the end of the first meiotic division, we have secondary gamete. Primary gametes are 46 chromosomes, double structure, 4 and DNA material. Secondary each have 23 chromosomes and which are also double structured and 2 N genetic material in each daughter cell. Is that right? Then when these secondary gametes go to the second meiotic division, simply these cells don't go into interphase, because in interphase DNA replication should occur, but they do not go into interphase. Cells from first meiotic division directly go into second meiotic division without replicating their DNA, but what they do, double structured chromosome break apart at center, right, centromeres, and single structures or single chromatid go to the daughter cells. Each single chromatid from the double structured chromosome go to the cell, and 2 secondary gamete eventually make 4 final daughter cells. Any question you have? Diversity, genetic diversity is assured during these steps. Number one, genetic diversity occur for the next generation. Number one, due to crossing over, where genetic blocks of genes are exchanged. Number two, genetic diversity is further ensured by random assortment of maternal and paternal homologous chromosome to the daughter cells during first meiotic division. And in the end, third mechanism of genetic diversity come when, yes, genetic material from sperm and genetic material from the ovum fuse with each other. They are having different combination, and an individual comes with unique genetic makeup. Any question here? There's no question. Let's have a break.